The anatomy of larger animals, in comparison to rodents such as canines and felines, can aid in the study of the anatomy of humans. One aspect in particular, the brain. In these animals, many diseases are known to occur spontaneously, just as they do in humans. Naturally occurring diseases in canines and felines have a significant advantage for translational research in that they frequently demonstrate the same clinical signs and pathophysiology as the diseases in humans. Accumulating evidence in experimental models has shown that the central nervous system damage alters the migration patterns of endogenous neural progenitor cells. The location and characteristics of the normal canine and feline rostral migratory stream are now reported to facilitate the study of neural progenitor cells and stem cell therapy in large animal models of central nervous system disease. The rostral migratory stream of endogenous neural progenitor cells has not yet been identified in these larger animals. 7 scientists from the University of Pennsylvania and Philadelphia conducted a scientific study on how the RMS in the canine and feline brain can further aid in the studies of the human brain in regard to neurologic disorders. The research article entitled "The Identification of the Rostral Migratory Stream in the Canine and Feline Brain" was published in the journal PLOS ONE on the 10th of May 2012. The study used 5 colony-raised cats ranging from 1 to 3.5 years old and 5 dogs ranging from 1 to 5.5 years old. Each experimental animal, except for cat 1 and dog 5, was administered 1 dose of freshly prepared 5-bromo-2-deoxyuridine and saline per day for 4 or 5 days, and they were perfused 24 hours after the final dose. Cat 1 and dog 5 were administered 1 dose of 5-bromo-2-deoxyuridine and perfused 6 hours after the dose. The canine and feline brains were carefully removed and post-fixed in formalin. Sagittal sections were then prepared for the study. The RMS was located by the use of immunohistochemistry, nasal staining, and image capture and analysis. The RMS in the canine and feline brains was located by the scientists, and an initial characterization was made from the nasal stained sagittal sections. The RMS tract corresponds closely to that of the RMS in a human brain, but is much more longer in proportion. 5-bromo-2-deoxyuridine immunoreactive cells were present throughout this tract. The RMS was also immunoreactive for many markers, and Luxelfast blue and CN paste staining indicated that myelin is closely opposed to the RMS along much of its length and may provide guidance cues for the migrating cells. In summary, this report identifying, mapping, and measuring the RMS in the canine and feline lays the groundwork for future studies to better understanding the involvement of endogenous neural progenitor cells and the pathogenesis of neurologic disorders.